Assalamualaikum all. Today we are going to start our unit 4. We are already done with the first 3 units. So today we are going to start with the unit 4. That is the plant growth regulators or phytohormones. So what are phytohormones or plant growth regulators? These are the organic compounds produced other than the nutrients like the calcium, magnesium, and what they do, what's their job? They promote, inhibit or modify the growth in the plants, and these are produced in very low concentrations in a plant, and they have a direct role in the metabolic activity of a plant. So next if we see in plants, we have 5 types of the growth regulators or phytohormones. Number one, auxins. Number second, we have cytokinins. Third, we have gibberellins. Number next, we have abscisic acid, and then we have ethylene. There are others also like jasmonic acid. I'll be discussing those also, but these 5 are the main plant growth regulators or phytohormones in a plant. So number first we'll be discussing the auxin. Auxin was the first hormone to be studied in the plant. It does what? Its job is it plays an important role in the growth and development of the plant. It does stem elongation, it plays role in apical dominance, root initiation, fruit development. We'll be discussing these also. So coming to the discovery of the auxins, first the step towards the discovery of the auxins was led by Charles Darwin and his son Francis Darwin. What he did, he performed an experiment on canary grass. Canary grass is a plant. He observed what he did. He put a source of unilateral light, which unilateral light means light which comes in only one direction. He observed what? That the plant starts bending towards the source of the unilateral light. Fine. He observed this, and he concluded in his experiment that the substance which is responsible for making the plants to bend towards the source of the unilateral light lies in the tip of the plant. How he concluded? He performed different experiments. He cut the tip of the plant. He covered the tip with the opaque material. When he cut the tip, he did not see any bending. When he covered the tip, he did not see any bending. And when he covered the tip with a transparent cap, it showed bending. And when he covered the base, it showed the bending. So finally he concluded that the substance responsible which makes the plant to bend towards a source of the light lies in this plant tip by his experiment. And same experiment was also done by Boynton and Jensen in 1913, but Darwin and his son were the first to perform this experiment in 1880. Fine. So moving next, we see the main step towards the discovery of the auxins was done by this F.W. Wendt in 1926. What he did, he chose a plant Avena sativa or oats. You can simply say oats. Fine. What he did in his experiment, you can see he just cut the tips of these Avena sativa oats plants. He cut the tips and did what? Place these tips on the agar blocks. Agar blocks here we have, and he put these tips on the agar blocks. Later, what happens? He placed these agar blocks on the stems with no tips. Fine. And when he put these plants in the light, he observed that the bending takes place. So what he concluded? He concluded that the substance which was present in the tips diffuses into the agar block. Fine. He concluded that the substance present in the tip of the plant diffused into the agar block, and this agar block when placed on the decapitated plant showed bending. The stem showed bending. So the Wendt was the first who isolated the auxin. He discovered and isolated the auxin. Fine. And the name auxin was given by Kogel and Hagen Smith in 1931. The name auxin was not given by F.W. Wendt. It was given by Kogel and Hagen in 1931. Auxin means to grow. To grow. Fine. So there were the scientists Darwin, his son Francis Darwin, then came the F.W. Went, and then came Kogel and Hagen Smith. They named these. So this was all about the discovery of the auxins. So next we are going to see where these occur in the plant, occurrence and distributions of the auxin in the plants. It occurs universally in all plants. Where there is active growth, there is auxin production. जहाँ भी plant में active growth होती है, auxin will be there. Growing meristems and enlarging auxins, organs produce auxin. Shoot apex produces much auxin than the root apex. Apical bud synthesizes more auxin than lateral bud. Apical bud, you know, which is present on the top, and lateral, which are present on the sides of the plant. Developing seeds contain more auxin than matured seeds. Apical bud synthesizes 6 times more auxin than the expanding leaves. Fine, I hope this is clear, occurrence and distribution. Next we come to the structure of the auxins. So here is the principal naturally occurring auxin in higher plants, and that is indole 3 acetic acid, IAA. It is the principal naturally occurring auxin in higher plants. Its structure is like this, and next we have indole 3 butyric acid. We have different types of the auxins in the plant, but the principal one is this indole 3 acetic acid. So the common feature you can see in these types of the auxins are they have a unsaturated planar aromatic ring. This is a ring, aromatic ring, but it's unsaturated. It has double bonds and it's planar. So what is a common feature? You can say they have planar, unsaturated, aromatic rings and have a carboxyl side chain. You can see here's a carboxyl group, here's a carboxyl group. So they have the carboxyl side chain. So one thing you see in common to all auxins is that they have the planar unsaturated aromatic ring structure with the carboxyl side chain. Fine. Here we have 2,4-dichlorophenol acetic acid. Again, it has a planar aromatic unsaturated ring and a carboxyl side chain. We have 2,4,5-trichlorophenol acetic acid also. Fine. So these are 5 types of the auxins. So coming to the role of the auxin, what auxin does? Auxin does the following activities in the plant. It does the cell division and cell elongation. Promotes flowering, tropisms like phototropism, gravitropism. I'll be explaining these. Like it makes a plant to bend towards the light. Prevents abscission of the fruits and leaves. What is abscission? Abscission means falling of fruits and leaves. Abscission means falling of fruits and leaves. Fruit development, apical dominance. I'll be explaining this also. Root initiation and development. And cell differentiation. You see, high auxin concentration stimulates differentiation of xylem. If we have high concentration, if a plant have high concentration of auxin, it stimulates the differentiation of xylem, and low auxin concentration induces the phloem differentiation. So the differentiation in plants is done by the auxin. Cell differentiation is done by auxins. So here we go about the role. Like apical dominance, I told you about the apical dominance. You know, apical dominance is a process in which the apical bud, this is apical bud, it dominates the growth of the lateral buds. These are the lateral buds. Here these are the lateral buds and this is a apical bud. When the apical bud dominates the growth and development of the lateral bud, this process is called as apical dominance. And how is this apical bud dominant? It synthesizes auxin. Auxin produced in the apical bud inhibits the lateral bud growth. Fine. So how it was experimentally proved? When the apical bud was cut, the lateral buds showed the growth. When the plant was decapitated or the apical bud was removed, the lateral buds showed the growth. So it was proved experimentally like this. And when externally lanolin and IAA, indole acetic acid, was given to the plant, which is an auxin, what happened? It suppressed the growth of the lateral buds. Fine. When it was externally given, The apical bud was not placed, but the auxins were given externally. The lateral buds were suppressed. The growth of the lateral buds were suppressed. And what happens? Auxin transport inhibitor applied allows lateral buds to grow. And when auxin transport inhibitor was given, the inhibitor which blocks the transport of the auxin was given, means the auxin is not transported to other parts of the plant. The lateral buds show the growth. So apical dominance was proved in this manner. So next I told you it prevents the leaf abscission. You know, the leaf abscission is brought about by the hormone, gaseous hormone ethylene. So if it was seen when there was high concentration of the auxin in the leaves, it prevented the leaf abscission. It prevented the leaf abscission. Fine. Abscission means yellowing and then falling of the leaves. First the leaf turns yellow and then it falls off. So if auxin was present in much higher concentration in the leaf, it was shown that the abscission was delayed or prevented. And auxin promotes the development of the fruits. Fine. It promotes the development of the fruits also. And what does it do? Auxin promotes root formation, but it prevents the formation of the primary root. ये जो है primary root, इसका जो formation है, auxin prevents it. But what it does, it induces the formation of the lateral roots. Lateral roots are these, which develop from the primary root. This is the primary root, but auxin induces the formation of lateral roots. And if we take a stem cutting, for example, if we take a stem cutting from the rose plant and plant it in the soil, you can see after some days it develops new roots, and those roots are also developed by auxin. You can say auxin induces the root formation in the stem cuttings. It induces the adventitious root formation, lateral root formation, but not the primary root formation. So this was all about the auxins. So tomorrow we'll be discussing the gibberellins. So in auxins, what we did, we studied the distribution, we studied the discovery, and we studied the experiments related to the discovery of the auxins, structure of the auxins, and role of auxins, apical dominance. I hope I am clear. If there is anything, please let me know. Thank you so much. Stay blessed and stay safe.